Hello, my name is Jason Linville, and this is part of the SDR separation lecture. This part specifically is about preparing our samples for electrophoresis. We just got done talking about some concepts of DNA being negative and racing through a tube, just like a bunch of racehorses. Okay, in this portion, what I want you guys to focus on is describing how the amplified product is prepared. We're taking out that tiny tube, and you're actually going to squirt it into a different tube and add some other reagents to it, okay? Specifically, we're going to be adding formamide and size standard. So I want you to focus on what does the formamide do and what does the size standard do, okay? All right, so how do we prepare these? What we're going to do is we're going to get our PCR product out of the thermocycler. So again, we didn't get to do this in class in the spring of 2020, but your amplified product, it's in a tube smaller than your DNA extraction tube, but it contains your DNA, primers, polymerase, DNTPs. After you put it in a thermocycler, heats it up and down 30 times, you have 10 million copies of each of your alleles, okay? That is our PCR product, our amplified DNA, the tube that you remove from the thermocycler. Formamide is a chemical that we order from a company, okay? And formamide is just, it's a liquid, and I can't remember the CF3. That's not right. CH3 is there? Okay, who cares? You guys, that's a demonstration that you don't need to know the chemical formula for formamide. But if you look at formamide, it just looks like any other organic solvent or water. It's just a clear liquid in a jar, okay? And the formamide, the way formamide is different than water is water gives DNA the ability to form hydrogen bonds in the middle, and it sort of keeps the DNA in double-stranded form. This formamide actually lacks the hydrogen bonds necessary to form those hydrogen bonds, and the DNA, our ladder, will actually fall apart in the middle when it's in formamide, okay? So formamide is actually going to be able to keep our DNA into single-stranded form, okay? For the size standard is going to be, again, a reagent that we order from a company, and we're going to go over more about what the size standard is, but it is sort of a ruler. If you think of this as sort of a ruler, a way to measure the DNA, and we're going to put this sort of internal size standard, we're going to put this into every single sample to help us measure how large those products are, okay? It's sort of like taking a standard horse and no, we're not going to talk about horse race. Okay, so to prepare the tube of the PCR product, we're going to mix formamide size standard. We're going to add that to the PCR product, okay? We're also going to prepare a tube of allelic ladder, and the allelic ladder sort of serves as a fake sample in addition to our, it's sort of like a PCR product, but it's synthetic PCR product. It has all of the alleles in it. And we're gonna, again, use that as sort of a standard to compare our samples to, okay? I want everybody to recognize that size standard is something, a reagent we purchase, we buy, we use it to help determine the size of our alleles. Allelic ladder is something we purchase and buy. We help use it to determine the size of our alleles. It's very easy to get size standard and allelic ladder confused. Okay, so we're going to go over what each of those are, and each of them does a different thing. Okay, so just pay attention and be forewarned. Okay, I need to be able to differentiate size standard and allelic ladder. All right. Okay, we've changed our horses to alleles. Okay, and remember, in the end, if we were to amplify, if we were to amplify just one area of DNA, then in our tube, we may just have like the 7 and the 9. Okay, what these represent is this 7 here, we actually have like 10 million copies of this fragment of DNA, and we'd have 10 million copies of this fragment of DNA if the person was a 7 and 9. And they'd all be mixed together in that amplified DNA. They all start in our preparation tube at the starting line. But when we start racing these alleles down the tube, all 10 million of the 7s are going to travel together and travel a little bit faster than the 9s. Okay, so again, what this image is representing, where if we have DNA, it's not a mystery as far as which horse is going to win the race. We know which of these amplified products is going to finish first. Okay, so in the chat box, if we race, okay, the 7 allele, 9 allele, 10 allele, and 12 allele down a tube, which one is going to finish first? Which one is going to reach the detector and the finish line first? The 7, 9, 10, or 12. Okay, and you guys are correct. It's 7. It's smaller. Has to pass through that polymer. It can do so quicker. Okay, it's the quarter on the Plinko board, if you want to think of it that way. Okay, all right. So there's not just one fragment. Again, what this drawing is representing is those 10 million pieces, and they all travel together because they're all the same size. So they all travel like a cloud down the tube. Okay. Also remember, for each person, there's only going to be 2 fragments in each tube. So here I've changed it a little bit where we have this person is a 9 and 12. Okay, so if we're just amplifying one location of DNA, then we have 10 million 9s, 10 million 12s. Okay, when we race them down the tube, okay, they're going to hit the detector. Okay, the other thing that we want to change here is David Caruso is not judging a horse race. Okay. This, if you'll notice this yellow line, this represents our capillary. That's where they travel. And then eventually when they hit the capillary, there's going to be this detector window, and that's going to be the part that's sort of closed off and inside the machine. And in that detector window, the coating of the capillary is burned off. So the DNA is actually passing through like a clear glass tube. And when it hits that clear glass tube, a laser is going to zap it, and a camera is going to be just taking a picture. Okay. The camera's just sitting there going, do I see any light? Do I see any light? Do I see any light? Okay. And if DNA passes that detector window, it is going to give off light, which we'll cover in the STR detection lecture after quiz number 3. Okay. So don't worry about that. Just know that the camera can see the DNA when it crosses that detector line. Okay, so we begin our race, the 9 beats the 12, okay, and we wind up with, again, this graph, this electrophoregram is what it's called, that's going to show us the 9 took about 19.5 minutes to get to the end, the 12 took about 23.6 minutes. Okay, so each of these is going to be associated with the time it took for the DNA to get from the beginning of the capillary to that detector window. Okay. And this is what we're going to see when we start to analyze the data. We're going to look, the computer shows us, because it records, okay, there was no DNA hitting the detector window. There was no DNA hitting the detector window. But at 19.5 minutes, oh, we saw DNA. Okay. And then we didn't see any more DNA. We didn't see any more. And then at 23, oh, we saw DNA. Okay. And starting out, what the instrument records is the number of peaks and also the time that it took the DNA to reach the detector. Okay, so we have peaks and we have time. Now, you would think, oh, 19.5 minutes. I go and I look at my chart and I see allele 9 equals 19.5 minutes. And I go, oh, allele 9 took 19.5 minutes. So therefore, this must be allele 9 because it took 19.5 minutes. The problem is, and the reason why this lecture gets a little bit complicated, is because on a different day, on a different instrument, when the polymer is different, when the buffer is a little bit different, your DNA may be exactly the same. It's not the DNA that changes. It's the instrument and the reagents that are running the instrument. So some days, 19.5, it may actually take 18.5 minutes for the 9 allele to get there. For this, it may take, 22, okay? Some days those peaks run faster, some days they run slower. Because of that, we can't just look at the time and assign the allele number, okay? We actually need to take the time to convert this number twice, okay? Once into base pairs, and then we can convert it into the 9. It's a 2-step conversion process. So it's not even a 1-step conversion process where we run some type of standard and that will allow us to say, today 19.5 equals 9. No, it's a 2-step conversion process. And that's why this is a little bit more complicated. So what I want you to understand is right when the samples are done running, okay, you have peaks, and each peak has a time associated with it. And what we're going to try to figure out and try to be able to explain is how we change that time into allele. Okay. All right. So we are going to, did I not put this? Hold on. I need to go back because I think did I forget something? Explain the purpose for adding formamide, the purpose for adding size standard. Okay. We covered that. We covered this. I'm going to stop this mini lecture. And I'm going to talk about correctly measuring alleles, which I believe is going to use the rest of the thing. Yes, okay. I couldn't decide whether to divide this into 2 or 3 lectures. I'm stopping this lecture because it's the end of that mini lecture.